into describing each of these cell types, basophils are one of our granulocytes. They represent about 1% of the total leukocytes. They are non-phagocytic, meaning that they do not have the ability to engulf or to destroy in that way that other cell types do. They do defend against large parasites by releasing those toxic granules. So I mentioned earlier that the granules that are present inside the cell, which you can see here, are just these toxic substances that are aimed at destroying pathogens, invaders, and usually those are large parasites here for basophils. Now, to kind of distinguish here large parasites versus small parasites, right? What is the difference? So a large parasite would be like mice or lice or mites. And then we'll talk about the eosinophils, which usually deal with smaller parasites on the next slide. Now, because basophils are usually releasing histamine and heparin, we typically see a lot of basophils as a part of allergic reactions. So if you're allergic to nuts or bees or anything like that, you will see a lot of basophils. They will aggregate to the site and typically release histamine and heparin, which are really important mediators of an immune response. We spoke about histamine. Anyone remember histamine's role in mediating an immune response? We talked about it as an example with capillaries. So we said that histamine makes capillaries leaky, right? It increases the permeability of capillaries. And so we have substances that can move easily from the blood, which is where most of these leukocytes are, out into the peripheral tissues in order to start attacking those invaders. We also have heparin. Heparin helps with vasoconstriction, as well as heparin, I'm blanking. Okay, so histamine helps with the permeability of the capillaries, and heparin helps with the solubility of blood. It's going to help thin the blood to help those mediators get out into the tissues to help with that response. Okay. All right, eosinophils. So eosinophils represent about 1 to 4% of leukocytes. So again, not a very significant number. These do have the ability to phagocytose, but that is not their main role. Their main role is in defending against parasitic invaders. So these are smaller parasites, things like amoeba, things like protozoa, which is malaria, is an example of that, like hookworms, tapeworms, those kind of smaller parasites are where we see eosinophils present. And they are one of our granulocytes, so we do see those cytotoxic granules that help with that immune response. Neutrophils. So neutrophils are probably the most important and most abundant leukocyte. These are about 50 to 80% of the total leukocyte count. They have the ability to phagocytose, so engulf bacteria. They typically circulate in the blood for about 7 to 10 days, and then they can migrate out into the tissues where they help to mediate an immune response, usually by phagocytosis. So typically when you have pus, if any of you have ever seen pus at the site of an infection or injury, this is basically lots of dead neutrophils, right? Neutrophils are a sign that an immune response is being mediated. So that's why pus is always considered as a good thing, right? If you don't see pus, then that is typically signs, an ominous sign that there isn't an immune response, and you're probably heading to gangrene or some type of other unhealthy response to injury, okay? So the increased number of neutrophils that we see, both in the blood and out in the tissues at the site, is an indicator of an immune response being mediated. Okay, monocytes. Now monocytes, we mentioned earlier, are the precursors to macrophages. Macrophages are also important in terms of that pus response to tissue injury or to debris or bacteria or infection. Monocytes represent about 5% of our leukocytes. They also have that phagocytic ability, and they can leave the blood and become macrophages when they're in the tissue. So it's pretty much the same cell. In the blood, it's called a monocyte, and when it extravasates out into the tissue, it's called a macrophage. Okay? It's the biggest of our leukocytes, although the neutrophils are the most abundant. Okay, so make sure you're making that distinction. Now, we have two types of macrophages. They can pretty much stay where they are at that site of injury or infection. We call those fixed macrophages, or they can continue to circulate and scavenge the area for additional infections, and we call those wandering macrophages. All right, so those are pretty much all of our main cell types. Now we'll move on to speak about lymphocytes. Now, lymphocytes is a whole nother group of cells in and of themselves, and they're further divided into other types. They represent about 30% of our total leukocyte count, and they're most of the cells that are found out of the blood and in the tissues in the interstitial space. Now, as I mentioned earlier, one way to kind of indicate or separate or distinguish lymphocytes is this really large nucleus, which takes up most of the cell. So we have three types of cells here. We have B lymphocytes, which are called B cells, T lymphocytes, T cells, and then null cells, which are sometimes called natural killer cells. And these are the cells that I mentioned earlier do have the ability to differentiate into cells that can acquire granules. So while the main lymphocytes don't have granules, you may see null cells associated with granules. Okay, so B cells are associated with antibodies, and pretty much when a B cell comes in contact with an invader or a pathogen, it will differentiate into what is called a plasma cell. And our plasma cell has the ability to secrete an antibody that is the exact response of a specific antigen. Okay, so let me slow down here. Let's describe what an antigen is. So an antigen is anything that the body detects as being foreign. Most viruses, like coronavirus, or most bacteria, have something on their cell membrane that the body can identify as being not of self, okay? And that is what we call an antigen. So when a when a B cell comes in contact with an antigen, it will differentiate itself into a plasma cell that will secrete the exact antibody in response to that antigen. Okay, and so that is how we can mark different cells or different viruses for destruction in a specific way to whatever that invader is. Now, the other cells that are involved here are T cells. So when B cells do the seeking and identifying and tagging, T cells will come in and do the actual destruction. Okay, so T cells are involved in directly attacking or directly damaging that pathogen. They can be brought about if there's any type of infection, if there's any type of cancer, or any other presence of inflammation or transplanted cells. This is kind of a unfamiliar territory, right? So anything that the body identifies as foreign, which may be some type of transplant or a foreign substance, will elicit this response. Now they differentiate into what are called cytotoxic T cells, and these cytotoxic T cells are the ones that are actually doing the destroying, okay? Now this takes a couple of days. This is why if you have a virus or if you have a cold or flu, you will take a while to mount that response, right? It takes a while to get these processes warmed up, for B cells to identify the pathogen, to tag the pathogen, for T cells to differentiate into those cytotoxic killer cells and come in and start that immune response. And what the cytotoxic T cell does, it has these projections on its cell membrane, and it basically bores holes in the cell membrane of that pathogen. So if it's a bacteria or if it's a virus cell, it'll basically poke holes in the cell membrane of that target cell, and that is how we can lyse the cell or rupture the cell and destroy that pathogen. Okay, so the kind of big picture here is that B cells find and tag, and T cells kill and destroy, okay? And then null cells. Now, null cells are what we call natural killer cells. These are very specific to viral infections. They help to identify and tag and attack viral infections or viruses and rupture these cells in a very, very similar way as those cytotoxic T cells. These are much more fast-acting than cytotoxic T cells, and they're usually involved in that really early immune response in terms of identifying and killing these invaders. Okay, null cells also have the ability to further differentiate, as I keep mentioning. They can differentiate into what we call dendritic cells, and dendritic cells have a different role, but they do acquire granules as a part of that role. Okay, any questions so far? Any questions? Yep. Yep, exactly. So they can either be natural killer cells or they can differentiate into dendritic cells. Okay, any other questions? Okay, so let's speak about how we actually synthesize these leukocytes. Now we know that all cells of the blood come from this one same hematopoietic precursor cell or the stem cell, so the same precursor that gives rise to erythrocytes and eventually megakaryocytes, which become platelets, also give rise to all of our leukocytes, okay? And that's a really important point to make in terms of the clinical application of leukocytes and this precursor cell. So if we have excess production of erythrocytes, we have a decrease in the production of leukocytes, okay? So because they're all coming from that one precursor cell, it's really important to understand the moderation or the proportion of these cell lineages, right? If we have an infection going on, we're gonna decrease our red blood cell production and vice versa, okay? Now they develop to maturity, usually in the bone marrow, but T lymphocytes are a little bit different. They migrate to the thymus. Anyone know where the thymus is? Where's the thymus? Do you have a thymus at this age? So the thymus is this fatty gland that is usually present before puberty, right? It's anterior to the heart, and it's usually really large in infants. And by the time you're past puberty, it completely involutes because the thymus has one job. It's responsible for the development and the maturation of T lymphocytes into mature T cells. Okay, so that's kind of the function of the thymus and the location. So the T lymphocytes will go to the thymus and then develop to their full maturity. This is under the control of colony stimulating factors and interleukins, which are basically fancy words for saying growth factors that stimulate the maturity of these T lymphocytes.